Cell cycle and cell division. Short notes. Introduction. All cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. Growth and reproduction are characteristics of cells, indeed living organisms. Such cycles of growth and division allow single cell to form a structure consisting of millions of cells. Cell cycle. It is the sequence of events by which a cell duplicates its genome, synthesizes the other constituents of the cells, and eventually divides into two daughter cells. Cells growth result in distributing in the ratio between the nucleus and cytoplasm, and therefore became essential for cell to divide to restore the nucleocytoplasmic ratio. Cell growth in terms of cytoplasmic increase is a continuous process. Duration or cell cycle can vary from organism to organism, also from cell to cell, type of cell. Each cell duration 90 minutes, human cell duration 24 hours. Phases of cell cycle. Interphase and M phase. Interphase, called the rest phase, cell preparation for division by undergoing both cells and growth. DNA replication is an orderly manner. It lasts for more than 95% of the duration of the cell. M phase, actual division process, starts with nuclear membrane karyokinesis, eventually ends with division of cytoplasm cytokinesis. Interphase. Interphase is divided into three phases: G1, S, G2. Some cells do not further exit G1 phase to enter an inactivate stage called quiescent stage or G0 of the cell cycle. Cells of the stage remain metabolically active but no longer proliferatory unless called on to do, depending on requirement of organism. Example: heart cells. G1, interval between mitosis and initiation of DNA replication. Cell metabolically is active and continuous, grows but does not replicate, is a DNA. Most of the cells organelle duplicate. S. This phase marks the phase replication and chromosome duplication. Amounts of DNA per cell doubles, but there is no increase in chromosome number. In animal cells, centriole duplicates its cytoplasm. DNA replication begins in nucleus. G2, protein are synthesized in preparation of mitosis. In animal cell, mitotic division is only seen in diploid somatic cell. Plants can show mitotic division in both haploid and diploid cell. Most dramatic period of cell cycle. Chromosome number in parent and progeny cells are same, hence are called equigeneal division. Then M phase, mitosis phase, karyokinesis and cytokinesis. Karyokinesis consists of four phases: prophase, metaphase, anaphase, telophase. Prophase. First phase follows S and G2 phase of interphase, marked by initiation of condensation of chromosomal material which becomes untangled. Centrosomes start to move towards opposite poles. The completion of prophase can be marked by chromosome condense to form compact microsome, mitotic chromosome. Each centrosome reach at pole and radiate out microtubule called esters. The two esters together with spindle fibers form mitotic apparatus. At the end, prophase cells do not show Golgi apparatus, ER nucleus and nuclear envelope. Metaphase. The complete disintegration of nuclear envelope marks the start of metaphase. Condensation of chromosome is completely and can be observably clear under microscope, and morphology of chromosome is most easily studied. One chromatid of each chromosome has two sister chromatids which are held together by centromere. All the chromosome coming out to lie at equator. One chromatid, each chromosome connected by its kinetochore to spindle fibers from pole, and its sister chromatid connects by kinetochore to spindle opposite pole. The plane of alignment of the chromosome and metaphase is referred as metaphase plate. Then anaphase. Centromere of each chromosome split simultaneously and chromatin separate. Two daughter chromatids, now referred as daughter chromosomes, begin their migration towards two opposite poles. The centromere of each chromosome remain directly towards pole and arms trailing behind. Telophase. The chromosome decondense and lose their individuality. Nuclear envelope develop around chromosome cluster at each pole, forming two deuter nuclei, nucleolus and Golgi apparatus, and ER reform. Then cytokinesis. It is division of cytoplasm at the end of which cell division gets complete in animal cell. It is achieved by appearance of furo and plasma membrane. In plant cell, cytokinesis is achieved by wall formation, which is center of the cell and grows outwards to meet the existence lateral walls. The formation of new cell walls begin with the formation of simple precursor called the cell plate that represent the middle lamella between wall of two adjacent cells. When karyokinesis is not followed by cytokinesis, as the result which multinucleated condition arises, leading to formation of syntium, example liquid endosperm in coconut. Then kinetochore is disc-shaped structure at the surface of centromere, serves as site of attachment of spindle fibers. Then what are the significance of mitosis? Mitosis usually results in identical generation complement in daughter cells. Growth of multicellular organism. Cell repair. Mitotic division is apical and lateral meristem. Result continuous growth of plant throughout the life. In some lower plants and some social insects, haploid cells are also divided by mitosis. Cell cycle and cell division. Part 2. Meiosis. A special kind of cells division that reduces the chromosome number by half result in production of haploid daughter cells. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organism, whereas fertilization restores the diploid phase. Meiosis involves in two sequential cycles of nuclear division, meiosis 1 and meiosis 2, but only single cycle of DNA replication. Meiosis 1 is an initiation after parental chromosome has replicated. 4 haploid cells are formed at the end of meiosis 2. Then the M phase, then the meiosis 1 phase or reductional phase. Meiosis 1 or reductional phase. First there is prophase, then metaphase, then anaphase, then telophase. Then first is prophase 1. Prophase 1 have leptotene, zygotine, pachytene, diplotene and dikinases. Prophase 1 typically longer and more complex when compared to prophase of mitosis. Leptotene. Chromosome became gradually visible under the light microscope. The compaction of the chromosome continues throughout leptotene. Zygote. Homologous chromosome called synapses. Synapses is accompanied by the formation of complex structure called synaptimal complex. The complex formed by pair of synapsine homologous chromosome is called bivalent or a tetrad. Leptotene, zygotine are relatively short-lived compared to pachytene. Pachytene. 4 chromatid of each bivalent became distinct and clearly appear as tetrad. Appearance of recombination nodule at the site of where crossing over occurs between non-sister chromatids of the homologous chromosome. Crossing over is exchange of genetic material between two homologous chromosomes. It is an enzyme-mediated process. Enzymes involved is called recombinase. Crossing over leads to recombination of genetic material, which is completed by end of pachytene, leaving chromosome linked at site of crossing over. Then diplotene. Beginning of diplotene is recognition by dissolution of synaptimal complex and tendency of the recombination homologous chromosomes of bivalent to separate from each other except at the site of crossover. The X-shaped structures site of crossing over where are called chismata. In oocytes of some vertebrates, diplotene can last for months or years. Dikotene. Diakinases. This is marked as a terminalization of dioecious metachromosome are fully condensed and meiotic spindle is assembled to pair of homologous chromosome. By separation, by the end of dikinesis, the nucleus disappears and nuclear envelope outbreaks. Dikinesis represents transition of metaphase. Then there is metaphase 1, bivalent chromosome align on the equatorial plane, double metaphasic plate, microtubule from opposite poles of spindle attached to kinetochore of homologous chromosome. Anaphase, homologous chromosome separates while sister chromatids remain associated at its centromere. Telophase 1, the nuclear membrane and nucleolus reappear. Cytokinesis follows and it is called dyad of cell. Interkinesis. It is a stage between meiotic division and generally short-lived. No DNA replication. It is followed by prophase 1. Prophase 2. Meiosis 2. Equinal phase resemble a normal mitosis. Prophase 2, it is initiated immediately after cytokinesis. The nuclear membrane disappears at the end of prophase 2. Chromosome became compact. Anaphase 2. Prophase metaphase. Metaphase 2. Chromosome align at equator. Microtubule from opposite poles of the spindle get attached to the kinetochore of sister chromatids. Then anaphase 2. Splitting of centromere around chromosomes to move towards opposite walls of the cells. Shortening of microtubule attached to the kinetochore. Then there is telophase 2. Meiosis ends with telophase 2. Two groups of chromatids once again get enclosed by a nuclear envelope. Cytokinesis follows as resulting formation of tetrad cells. Then you have meiosis 2. Stages of meiosis 2. Meiosis 2 have 4 stages: prophase 2, metaphase 2, anaphase 2 and telophase 2. Significance of meiosis. What are the significance of meiosis? There are many significance such as conservation of specific chromosome number of each species is achieved across generation is sexually reproducing organism paradoxically result in reduction of chromosome number by half. Increase genetic variability in population of organism which is important for the process of evolution. Thank you.